Hi, this is Metabolism for Microbiology, and we're going to be following the structure of Dr. Sung's lectures. So let's go ahead and get started. And what we're going to basically be doing is focusing in on the simple concepts, or rather a simple version of these concepts that might seem kind of complicated. So to begin with, let's discuss monomers and polymers. So you've got think of mono, right? Single. Poly means many. Mono is like bricks, basically, little building blocks. You've got these simple sugars, and when you put them together, they form complex sugars, which are polymers. And these are like your starches. So you think of, like a brick wall or something where you've got many parts. These are all individual parts, these monomers, and they're coming together to build something more complicated. So like with glucose, we have a monomer. When you have two together, two monomers together, maybe they're bound by an oxygen or something, that would be called a dimer. And then when you get lots of these together, right, over and over, et cetera, that's a polymer. Okay, so we have these two really important terms, anabolism and catabolism. And so with this understanding of monomers and polymers, hopefully that can help us understand kind of what these terms mean. So with anabolism, we are building things up. We are building that wall. So taking all these individual parts and putting them together, right, with mortar to make these bricks bound together and become one more complex structure. That is anabolism, turning monomers into a polymer, or bricks into a wall. This is an endergonic reaction because electrons are moving in. You can think of electrons, and I'll continue to use this analogy, as mortar, right? So what goes between the bricks? What keeps everything together? It's electrons. Electrons, if you think about it like planets and moons, right? Like if these are atoms, maybe we'll have a water molecule or something here, okay? So you've got your little hydrogens and your oxygen and your hydrogen. What keeps these things from being just individual structures floating around on their own? Why is this one unit? Why do we have a unit called H2O? Well, it's because these electrons are orbiting very quickly. In fact, so quickly that it becomes like a rope. And it's almost like they're in several places at once if you were to just observe it in real time, because they're just moving so incredibly fast. What's going to happen is, and we'll come back to this later on, but this moon, so to speak, right, this electron is going to spend more time around the larger structure. It might orbit it a few times and then jump out here, and then orbit it a few times and jump out here. And so molecules like, or atoms like oxygen. Oxygen is very electronegative, which is kind of what I've been equating to gravity here, right? Like it's a planet. It's called electronegativity, and because it's more electronegative, it's more attractive to electrons. So that's why the electron is spending more time out here. So keep that in mind, and we'll come back to that. So when we're building up structures, the electrons are keeping it all together, right? The mortar. So this is an endergonic reaction because we are adding something to it. The electrons are going in, right, from somewhere else. We're adding in mortar, endergonic. By contrast, an exergonic reaction in catabolism, you think of like an explosion, right? If this were to explode, electrons are leaving in that case. That would be an exergonic reaction, right? An explosion. Or a catastrophe, right? You think buildings are destroyed in catastrophes. Well, that would be catabolism. Okay, so polymers turn into monomers in that case. We're moving from the wall down to its individual parts being unique again, and individual, because there's no mortar keeping them together anymore. There's no electrons. So in this, we are losing electrons. This is an exergonic. They're exiting, they're leaving, right? Okay, now here is a really important thing that people get confused a lot. You would think that this would be a reduction reaction, and it's actually not. It is an oxidation reaction, because reduction is not referring to the complexity of the structure, right? You'd think this is like reducing it. Don't think that. Reduction has to do with the charge, okay? Because electrons are negative, right? So if you have more electrons, you're building something up, you have more mortar, it becomes reduced in the sense that it has more charge, more of a negative charge, I should say. So as we're adding electrons in, right, we've got all these little bricks, these monomers, like maybe glucose, right? And then we want to bind them all together and turn them into a complex structure. You know, we want to build something with them. We're going to have to bind them together. And the way we're going to do that is with negative electrons. And these electrons are going to bind it all together and tie it all up so that it ends up looking something like this, right? We need negativity to be able to bind things together. Our mortar is negative, okay? So that's why you think reduction, like on a number scale, it's going down, it's becoming more negative, which means that it's being built. Something more complex is happening. So with anabolism, we're going through reduction reactions. With catabolism, we're going through oxidation reactions. I'm going to quickly explain to you why this is called oxidation, since I explained reduction. Oxygen is very electronegative. Remember, I described that over here. And so the electrons are going to spend more time around this. You could think of it like being electron hungry. And so before we really understood how electrons worked and everything like that, all of this stuff works, all we observed is that a lot of times things broke down when oxygen was introduced. Because oxygen is very electronegative, we found out later on, and so it's taking electrons away from other structures, right? So if oxygen comes along here, and, well, maybe let me draw it down here. Oxygen comes along down here and steals this electron away, right? It's pulling electrons away from it, getting this exergonic reaction, pulling the electrons out of this complex structure. Then it falls to pieces, right? So we call this oxidation because oxygen is a common instigator for these exergonic reactions, but it's not the only one. Anything can really cause an exergonic reaction, but the most common are nitrogen, oxygen, and fluorine. Those top right elements on your periodic table. Or I guess it's not flipped for you, maybe this way is your right. But regardless, it's that upper right corner is more electronegative. So just remember that reduction has to do with charge. So things are being built up. They are being bound together. We are doing anabolism. We are doing endergonic reactions, bringing those electrons in. That is reduction. Oxidation, you think it's stealing, picture oxygen stealing those electrons, and then it all falls to pieces because our electrons are our mortar. And so that's our exergonic, explosion, catastrophe, catabolism, right? So catabolism is our oxidation. Hopefully all the repetition helps it stick. So when we come to our redox reactions, or oxidation reduction is what that is kind of short for, look at, for example, NAD plus and NADH. these two molecules change back and forth between each other. And the difference is that, look at this extra part here, this extra, I believe it's a hydrogen and not standing for something else, but when it is reduced, see, reduction is this arrow moving this way, it's being built into a more complex structure. So there was hydrogen floating around out in the space like this, all over the place. Hydrogen. And one of these gets bound to this. Well, look, this is plus and this is plus. So they will repel each other, right? Normally, they're not going to touch each other. These are all going to be, there's repulsion happening here. But when we introduce an electron, which is negative, this now satisfies that space and then comes around here and satisfies this space, and it takes turns going around the two and ends up binding these together so that they become one unit. This is a reduction reaction. Now it's NADH, a more complex structure, because what's right here is that electron. Okay? When it's oxidized, we're going to lose that H. It's going to pop off because now it's repulsed once we lose that electron. So if something like, say, maybe oxygen comes along and steals that electron away, then we're going to be left with NAD plus and hydrogen protons. And these guys are not going to want to touch because they're both positive. Just like the two positive ends of a magnet, they repel each other, right? We need the electron to keep things together. So that's a redox reaction. Okay, so all of this is laying the groundwork. We're moving towards the production of ATP. Because our metabolism, if we really break it down into its simple parts, is we take in food and then we're able to do stuff, right? And we're trying to see the molecular biochemical level of how that's working. So now that we understand some of this stuff about catabolism, anabolism, reduction, oxidation, we can start to understand what's going on with enzymes, and then we'll move into glycolysis and citric acid cycle and all that stuff. So just give you an idea of where we're going with all this. So enzymes reduce activation energy. Activation energy. And activation energy is basically the amount of energy that you need to make something happen. So we're talking about chemical reactions like catabolism, for example. So if you look at this chart right here, this is an image. All these images are from Dr. Sung's slides, most of them anyway. So let's imagine that there's a ball right here, okay? And this is a hill. In order to get the ball to do something on its own, we need it to get to this point right here, right? And then it'll roll down the hill, and maybe we've got a wheel well there or something that could generate electricity from the ball falling down and turning it, whatever. Regardless, something is going to happen because of this fall. So in order for us to get to that point, however, we need to exert a certain amount of energy. And that energy that we exert to get to that point is known as activation energy, or E of A, energy of activation. So what an enzyme does is it comes along, like let's say, okay, the reaction that we're trying to do, the ball is an analogy. Let's say that the actual reaction that's happening is we're trying to break sucrose down into 2 glucose molecules, which is what it's made of. The only way that's going to happen is if we lose some electrons, right? We want an exergonic reaction to happen. We want some catabolism to happen. In order for that to happen and release its energy, like a little explosion, and break into its parts, we need to exert a certain amount of energy and kind of tug on it and pull it apart from itself. That is exactly what this enzyme does. This molecule of sucrose fits into the enzyme, which is specially designed for it. Look at the shape, it's perfect, right? And this enzyme fits, and now we have this, it's binding to the active site. And what it's doing here is applying tension on a molecular level, it's really like it's applying attention. It's connected all along here through charges. And what it's doing is it's tugging on it so that the chances that it'll spontaneously break apart are higher. And the way that that really works is that it's reducing the amount of energy required for it to have the catabolism reaction, which we want, right? So now instead of having to push it all the way up here, this steep, steep hill, we can exert less energy to get it up to the top of this hill, which means if we're doing this over and over and over and over to run this wheel well, we're going to be so much more efficient, and it's going to happen so much more in the space. I mean, if you imagine like doing this for a whole day, you know, you're going to be able to do this small hill way more times in that day than you are this big hill. And it's because there's less energy required to get to the point where it can have an exergonic reaction. Okay, so that's activation energy. And what an enzyme does is it lowers the activation energy. So this red line here is with the enzyme, and the green line is without the enzyme. Okay, hopefully that makes sense. Oh, and I made a mistake here. Sorry, it's glucose and fructose, not two molecules of glucose. Sucrose is made of glucose and fructose. Okay. All right, now there's another key element of enzymes, and that's that they often don't work on their own. They need to be activated by cofactors and coenzymes, which are essentially the same thing, except that cofactors are really typically they're elements, not complex structures. NAD or NAD and FAD, these are more complicated molecules. Magnesium, iron, zinc, these things are, they're just elements. They're inorganic, meaning they don't have any carbon in them. Whereas these do have carbon. They're complex structures with many elements in them. Okay, so cofactor, inorganic, coenzyme, organic, and larger and more complicated. And you can see here how they work. This substrate, which would be like our sucrose from earlier, right? This is the substrate, and here is the active site, and this is the enzyme, right? So in order for that substrate to fit into the active site, see how the shape is perfect right here, but over here, there's no way it's going to happen, right? So we need the coenzyme to fit into this space so that the substrate can fit into the active site. A cofactor does the same thing by changing the structure of the enzyme, because, you know, maybe it doesn't work so much like exactly like this one, but if you imagine iron over here having its own charge and it comes over and binds, or maybe it's on the inside of the enzyme or something, it's going to change the structure. So once it comes in here, it'll bend things around it, and this enzyme will change shape so that it looks the way it needs to with this proper active site. Same idea up here. Look at the cofactor, the coenzyme, fitting into its place. You know, this circle's not going to fit in over here, but it'll fit in here, and so it all works out. So we need these cofactors, coenzymes for it to work. Now, one last thing about enzymes is the concept of inhibition. So we have our competitive inhibitor, and we also have a non-competitive inhibitor. And the difference between those is really quite simple. This here is the active site. If there are two things competing for the active site, then the substrate that we want to get in there won't be able to, at least not as often, because some of the time this inhibitor it's really a competitor vying for the same spot. And if it's in here, you know, there's no room for this guy. So that's one way that it can happen. Competitive inhibition. With non-competitive inhibition, what's happening is kind of like the cofactor I was describing. When something binds, like in this space, for example, anytime something binds to another protein, it changes the structure. things binding together change the structure of them because we're putting tensions and there's all these charges involved. And these are really quite complex structures. And this is just a simplified representation. So when you have something like a non-competitive inhibitor enter this space, what it might do is actually change the shape of this enzyme so that now it looks like that, right? Maybe it would do something like that. Change the structure so that now the substrate can't fit in there, which is exactly, you could see what that's what's happening here, right? It's changing the structure of the active site by kind of this chain reaction of changing the structure of this enzyme. Okay, so we have competitive where it vies for the active site, and non-competitive where it's not binding to the active site, but it changes the shape of the active site by its binding. Okay. Now, feedback inhibition is where the product of this whole chain results in the inhibiting of the chain. So our substrate, by getting broken down, creates, through all these steps, an end product which makes more of the substrate unable to be broken down into this. So it's self-regulating. The more substrate that we have being broken down, the more that it's going to be inhibited. So this is really useful because let's say that you want to digest a certain amount of something. If the end product is an inhibitor, what'll happen is it'll digest it until it goes down to a normal ratio, and then it'll stop. It won't keep going until it's removed all of the substrate. It's just going to regulate itself so that the more of this that gets destroyed, the less of it can be destroyed. So I'll say that again. The more that this substrate gets catabolized, the more this end product stops that from being able to happen. And so this is called negative feedback or feedback inhibition. Okay. A positive feedback would be like if the substrate produced a cofactor or a coenzyme that comes along and makes it easier for the enzyme to catabolize these. And with that, you don't really have breaks. It's all gas, all gas, all gas, all gas, because the more of this that you break down, the more of this you break down. Whereas with feedback inhibition, the more of this you break down, the less of this you break down. Okay, so this is our breaks. This is a system that has breaks. Just keep that in mind. Feedback inhibition. Generally, it's more involved with non-competitive inhibition. Okay, so take a minute, pause the video, go walk around and take some breaths, do some jumping jacks, get some water or something, and then come back to it because we're going to put all these concepts together so that we can understand how the body turns food into energy. So glycolysis and the citric acid cycle are basically steps that are designed to break glucose down into parts that are going to be able to fuel the electron transport chain and oxidative phosphorylation, which is really our big ATP generator. So you see the NADH being produced at each of these steps. NADH is going to come back in later on, and FADH as well, in fueling the oxidative phosphorylation. So yeah, let's get into this. ATP, first of all, is kind of like a battery, and you can think of it like a fully charged battery. It has a lot of energy stored in keeping this together. And again, what's keeping all these parts together? Electrons. Okay, these are electrons keeping it all together. So when we lose one, you must also think that we are losing an electron, and it's an excited electron, that can go and be used to build other things, right? We need that electron to be able to build up stuff. And so this is kind of like a transporter of excited electrons, basically. ATP is very unstable. And it has to do with the structure of it and the fact that there's these negative oxygens on each of these phosphate groups. I'm not going to get into that so much. Just keep in mind that ATP is unstable. It wants to break down. This is a battery that wants to share its energy. And when it does, and we lose this phosphate, it now becomes known as inorganic phosphate. Sometimes it's symbolized by something like that, or something like that. Anyway, this is now adenosine diphosphate. We had triphosphate up here, 1, 2, 3, and now we have diphosphate, 1, 2. This is like your uncharged battery. Okay, and it has to get recharged by having an electron added back to it for it to get to its excited, unstable state. Whenever you have potential energy, you are considered unstable. So the classic example is somebody on a diving board, right? They're standing up here. They have potential energy because if they step off that diving board, they're going to have a lot of kinetic energy as they fall into the water. None of that has happened yet, and so they're considered unstable because eventually that's going to happen. Eventually they're going to come back down one way or another. And if they climb back down the ladder, that might be less catastrophic. And this is actually what the process of all of this is really working to accomplish as well, is it's breaking glucose down in small steps. Because if we went straight from glucose to ATP, or rather straight from glucose to cellular function, we would actually combust. There is so much energy, just like if you stepped off a cliff, you would just fall and fall and fall to your death. But if you start from the same height and you release that energy little by little, by going down a little at a time and then stopping, right? You could walk down a staircase that far completely safely and still release the same amount of kinetic energy. It's still the same distance, right? So glucose breaking down into pyruvate and then the citric acid cycle and all these steps, it might seem really convoluted and overwhelming at first. And just keep in mind that what's important about this is that we're doing it in steps so that we don't blow up. We can use some of that energy little by little through this really beautifully complex system. So we look at this picture and it's extremely overwhelming. You look at glycolysis and it's even more so. And I don't have a picture of that one here, I don't believe. Yeah, I do not. Okay, so what we're going to do is we're going to break it down in simple steps. So if you want to get out a piece of paper, pause it, and get a piece of paper and a pen. We'll do this together, okay? All of glycolysis is summed up in this transformation. We ingest glucose, which is a simple sugar, with 6 carbons. If you recall, if you've taken any other classes of this type before, this is the formula for glucose. 6 carbons, 12 hydrogens, 6 oxygens. I don't think that you need to memorize this, but the part that's important is carbon. Through this whole process, we're going to track carbon. Okay. Glucose has 6 carbons, so we'll put a 6 right there. And what it does in glycolysis is it breaks down into two molecules of pyruvate. Which each have 3 carbons, which just makes sense, right? 6 divided by 2 is 3. Because we've split it in half, essentially. This process, if you look at it, like on Dr. Sung's slides, it's very complex and a lot is going on. We have to add in some ATP, we get some ATP back out, but we're going to look at net values here, okay? So what we're gaining from all of this is every time we create pyruvate, we gain 2 ATP and 1 NADH. Yeah. 2 ATP and 1 NADH. Okay, and that'll make more sense if you look at the whole process. But for now, just know this much. Glucose to pyruvate, 2 ATP, NADH. Now, our pyruvate is going to each turn into acetyl-CoA. And this is known as the oxidation of pyruvate. So in this process, we go from 3 carbons to 2 carbons. How did that happen? Because we're only left with this step. There's no other thing out here. There actually is. It's CO2. There's the carbon right there. Okay, this third carbon, one of the carbons in pyruvate, got released in the form of CO2. And this is a perfect example of oxidation because look at what pulled it away, oxygen. And what's binding it all together is an electron that was keeping this all together. Right? So the electron that was keeping this carbon bound to the pyruvate was pulled away by some oxygens to make CO2, and with it, the electron that binds it. Right? So we're short an electron, we're short a carbon, and we're left with 2 carbons here. So if we're losing an electron, if we're losing a carbon, we're also losing an electron. And yeah, so let me retract what I just said. The electron that was keeping this together is not going to bind up CO2. It is actually going to bind up NAD+ and hydrogen+. And we're going to do that reduction reaction to form NADH. Same thing that happened up here in each of these steps, but here we're producing carbon dioxide, we're losing a carbon. Whenever we lose carbon and produce carbon dioxide, we are going to produce NADH. Okay, so keep that in mind because we're losing that electron. This electron that was keeping this up here is now down here with the NADH, keeping this bound to that. Right? Okay. Now, once we get to acetyl-CoA, it enters a cycle. And this cycle is going constantly. This is the citric acid cycle. So maybe we'll draw a line right here and right here. This is the oxidation. I think I said phosphorylation earlier, mistakenly. Oxidation of pyruvate. Because pyruvate was oxidized, it lost an electron, and NADH was reduced. Or NAD+ and H+ were reduced to make NADH. Okay, so now our acetyl-CoA is entering the citric acid cycle. And the way we're going to remember this one is with these numbers, 6, 6, 5, 4 4s. Okay, we have 4 groups of 4 there. So you got our 4 4s and then 2 6s and a 5. 6, 6, 5, 4, 4, 4, 4. And what's happening is this molecule right here will symbolize these carbons like this. And this molecule right here, which we'll symbolize as 2 carbons, are coming together. I mean, 2 circles symbolizing those 2 carbons are coming together to make a 6-carbon molecule. See, so this 2 and this 4 are coming together to make a 6. See, so this is where it's entering the cycle. And at each of these steps, you see going from 6 to 5, we're going to lose some carbon. From 5 to 4, we're going to lose some carbon. And so by the time we get down here to restart the cycle, we need this acetyl-CoA entering the cycle. And it'll look something like that. Okay, not super important what the names of these are. Not super important their structures. What's important is the number of carbons so that you can visualize the process. So then it comes down here and it basically just, some stuff changes that's irrelevant. You know, if you were taking a biochem class, you'd probably have to know about all the little details of this. But for microbiology, it's not such a big deal. The important stuff is when we see a change in carbons. So from 4 to 6, we know the 2 is entering in there. Okay, that makes sense. 6 to 5. What do you think is going to happen here? If we're losing a carbon, what form is it going to be in? If you said carbon dioxide, you're absolutely correct. It's going to lose some carbon dioxide, which means it lost an electron. Where is that electron going? It's the same thing that happened up here. We're going to produce NADH. Okay. 5 to 4. Same situation. If we're losing carbon, then we are creating NADH. Now from 5 to 4, you can remember this, that there's a party from 5 to 4, 5 p.m. to 4 a.m. And the reason that we call this one a party is because a lot is going on in this. And it's one of the only steps in our simplified version right here where 2 things happen at once. Because we are also producing at this step ATP. A lot is happening from the 5-carbon molecule to the 4-carbon molecule. And the ones that are late to that party are the FADH. We're producing FADH2 at this step. So parties from 5 to 4, and then late to the party is FADH2. Okay, so this is produced from FAD and 2 hydrogens being reduced to come together, right? To make FADH2. Okay. Nothing important happens at this step, just like up here. Okay, nothing really important happens there. There is one thing that is not so intuitive that happens at this very last step. The very last thing that happens before the citric acid cycle starts over is we produce an NADH without producing carbon dioxide. The electron comes from something else. We're not losing a carbon. I think it's actually a hydrogen. And if you see the complex structures, it makes sense. But again, you don't need to get into all that. Just know that this very last step produces NADH because the citric acid cycle as a whole produces 3 NADH per turn. So here we've got 3 NADH, 2 CO2s, because this last one doesn't produce one, and 1 FADH2. Up here, we had 1 NADH. Up here, we had 1 NADH. And we have a CO2. Okay. Same exact thing is going to happen over here, right? Acetyl-CoA, citric acid cycle. And so all of these things are really going to be doubled. So all of this is times 2. And all of that is times 2 because it's happening on this side as well. And if it helps you, write out this side. Do it on this side as well, and it should make sense. So you can see now the progression of these carbons and understand what is going on. So we go from 6 to 3. You're just going to have to memorize these results here. Also, something to keep in mind is that 2 ATP are produced at each of these steps, but in glucose prior to the point where it splits, in glycolysis, prior to the point where it splits, we are also investing 2 ATP at 2 different points in that chain. So if you start with glucose up here and you're moving down, we have to invest ATP twice before it even splits. And then we get our pyruvate with our 2 and 2, right? So because we're losing ATP here and gaining it here and here, that's a gain of 4 and a loss of 2, right? So our net gain altogether is plus 2 ATP. So just keep that in mind. We also have a minus 2 ATP situation going on here, just so you don't get confused and think that our net is 4 in glycolysis. It's not. Our net in glycolysis is 2 ATP, because 4 minus 2. Make sense? Okay. We have a total of 2 NADH as well, right? So our net results here, maybe I should change the color. Our net results, we've got 2 NADH here, 2 NADH here, 6 NADH here, for a total of 10 NADH being produced between glycolysis and the citric acid cycle. We have 2 FADH2. And we have 4, 5, 6, right? 1, 2, 3, yep, 6 CO2s. And our total with ATP, we've got 1 here produced and a total of 2 produced in glycolysis, a net of 2. And this is also times 2, right? Because this cycle is happening twice, 1 for each pyruvate. So per molecule of glucose, we also have 4 ATP produced. So this is really important. And the most important part of it is this section right here, because this is what's going to fuel oxidative phosphorylation, which includes the electron transport chain, which is going to produce the vast majority of our ATP. So now we will move into that step. But first, write this all down, walk through the process in your mind, and on the paper. Do this several times until you can do it from memory without looking at it at all. Here's a reference for you of basically what we did. See the 1 ATP in and the 1 ATP in and the 2 out and the 2 out. This is what I was talking about earlier. So our net is 2 ATP for all of this, and 2 NADH. And then here, 2 NADH. And then down here, 6 NADH. So you should be able to know by heart this last part and be able to work through the process and trace those carbons and see how the math makes sense, right? Losing that carbon into the carbon dioxide. Losing that carbon, it's going into carbon dioxide. Losing carbon, it's going into carbon dioxide. If you can do this from memory, you will answer several questions on the exam for yourself. And it'll also help you understand what your results are going to be, which is going to answer several more questions. And it gives you practice as you're writing this all out. If you're building something up, say verbally out loud, this is a reduction reaction. If you're breaking something down, say out loud, it's a catabolism reaction, or it's a exergonic or oxidation, right? All talking about breaking things down. The more you can repeat this and solidify it, the more it's going to benefit you on the exam. One thing to note is that bacteria, which are prokaryotic, these are kind of like mitochondria in and of themselves. And a lot of times they'll have a double membrane that they can do this process in. Like down here where it's talking about the cell membrane. Mitochondria exist inside the larger cell, right, with the nucleus and all the organelles and stuff. So just keep that in mind. This is kind of like a bacteria living inside. It's not, but the mitochondrion is kind of like its own bacterial thing. So just keep that in mind, the differences between where it's happening in eukaryotes versus where it's happening in bacteria. Okay. Here's another good point where you should take a breather, just a minute or two. Let that stuff sink in. You know, if you want to practice going through that whole process of seeing how our NADHs come out, do that. Then come back to this, unpause it. So our oxidative phosphorylation is this whole process right here. It's electron transport chain plus your ATP synthesis. Okay. The electron transport chain is just a part of oxidative phosphorylation. So let's break down this word, oxidative phosphorylation. Phosphorylation means adding phosphate. You're phosphorylating something. So if we have our ADP, right? And we phosphorylate it by adding a phosphate group, well, now we're creating ATP, right? That's an endergonic reaction, a reduction reaction, an anabolism of ATP. We just turned ADP into ATP, which is the whole purpose of all this stuff. We need energy to be able to do this. Okay. The oxidative part has to do with the fact that the electrons are being pulled down this chain by the electronegative oxygen. And you can see this oxygen is right here. We're moving from left to right. And we'll explain this in better detail in a minute, but oxygen is pulling the reaction along. Okay. Down here at the very bottom is oxygen. And each of these cytochrome complexes, which are same thing here, represented here by these rectangles, are the same things represented here. You don't need to know the names of these. At least I don't remember ever having to memorize them in Dr. Sung's class. And what's happening is NADH. Okay, just take a breather. If you're overwhelmed, stop it. Stop the video, I mean, and come back for a second, okay? NADH, which was produced from all that glycolysis and citric acid cycle, all of that was to get NADH and FADH2 to this point where they can do their job. And their job is to donate electrons. Their job is to break down. We built them up just so that they could be catabolized again. And those electrons that come out from this, it's going to leave this NADH turning into NAD plus and hydrogen, right? Because now there's no electron binding them together. So we're getting these electrons, and this electron excites and changes the structure of this cytochrome complex. And then once it changes the structure, it gets pulled down to the next structure. And then it gets pulled down to the next structure where it changes it again. And each time the structure changes, it pumps hydrogen across the membrane. So It's getting pulled down because this oxygen is pulling an electron from here, which makes this one want one, and so it pulls it from here, which makes this one want one, which pulls it from here, right? So it's a chain reaction being caused by this oxygen being electron hungry down here at this end. And the result, the simple version of the result, is that hydrogens are getting pumped through the membrane by these complexes as they change shape. So let's think of it this way. We have our membrane. We have our mitochondrion, actually. Let's start there. And we have two membranes, right? The outer membrane and the inner membrane. And what's happening in this space here, we've got our intermembrane space, and then we've got our mitochondrial matrix here. So we'll label the top here matrix and the bottom intermembrane space. And what separates them is this inner membrane right here, which is a double layered membrane, just like all, right? It's a phospholipid bilayer. Don't worry about that too much. If that sounds foreign to you, it's okay. And embedded into this membrane, again, we're talking about this membrane right here, is cytochrome complexes. And what NADH does, let me draw it a different way. Yeah, let's make it something like that. This is a cytochrome complex, or a representation of one. And we've got all this hydrogen protons, hydrogen ions, it's all the same thing, floating around in this mitochondrial matrix. And what's going to happen is this hydrogen will come over here and bind into the cytochrome complex. Remember, it's a hydrogen proton. A hydrogen is only has one proton. And so if it's positive like this, it has no electron, and hydrogen doesn't have any neutrons. So it's literally only a proton, and it's positively charged because of that. So when we talk about protons, we're talking about hydrogen, ions, it's all the same thing. Just keep that in mind. So when this proton comes down here and binds into this complex, and then the complex changes structure because of an electron, what happens is this, okay, imagine this same complex here, changing structure so that now it's like that, right? What it's done is effectively pumped this hydrogen across the membrane, which is why these cytochrome complexes are also called proton pumps, because they're pumping protons from the mitochondrial matrix into the intermembrane space. They are only able to do this because they are changing shape, and they are only able to change shape because an electron is jumping over and changing the structure. It's changing the charge, and there's this whole complex chain reaction that happens in here that changes the structure. If you ever played with those magnet kits as a kid, where it's like the little magnetic marbles and the little magnetic sticks between them, if you stick a powerful magnet on one end of your beautiful creation, it changes the whole thing because it's moving electrons from one end, and it's causing this whole reaction, right? So it causes a chain reaction, structure change. What you need to know is that an electron jumps over here, it changes the shape of this proton pump, which causes it to do the pumping action. Where did this electron come from? NADH. And so in donating this electron, again, now this is going to break into NAD plus and H plus. Well, how convenient is that? It breaks into H plus as well, increasing the concentration of hydrogen protons on this side so that we have stuff to work with, right? So isn't that just like beautiful? it's all set up perfectly the way that it needs to be. So this NADH is donating this electron, which is changing the shapes of these cytochrome complexes, which then pump hydrogens onto this side of the membrane into the intermembrane space. And so now what we're left with is actually a higher concentration. I know these look a little different. These are all hydrogens too, down here that I'm drawing. We're left with a higher concentration of hydrogens on this side of the membrane. Because of this pumping action. It's pumping it against its concentration gradient. And so we have something called chemiosmosis, which is basically chemicals want to follow, pretty much everything on a chemical level wants to move to an area of lesser concentration. So osmosis is like when that happens with water, right? Water moves into a space where there's less water. Chemiosmosis, we're talking about these hydrogen protons. They want, so to speak, to get back on this side. They want to move this way, kind of like flowing downhill, right? They've been pumped up the hill, and now they want to flow back downhill. And so, built into the membrane is a space for them to do this. And it's known as ATP synthase. And so what happens is these hydrogens, I'll draw you a little simple version right here. They basically go through this tube with the molecular equivalent of a water wheel and spin that water wheel, so to speak, and it acts like a motor. It's literally a molecular motor generating energy from the flowing of these hydrogen protons down their concentration gradient back to where they started. So as they flow, this wheel spins. And as it spins, the electricity or the electrons that are generated, it's kind of a complex process in and of itself here, but what you need to know is, like, think of it like a motor. When it spins, it creates electricity, or it creates usable electricity, and it charges the battery. Okay, we spin this motor like a water wheel and create electricity, usable electricity, and that energy charges ADP and inorganic phosphate to come together in the form of ATP, which now wants to go and give that energy. This process right here, this tiny little process, is what everything is leading up to. The entirety of glycolysis, citric acid cycle, electron transport chain is all leading up to this moment so that we can produce a large amount of ATP that our body needs to do its cellular functions. So this ATP synthase, our protons are flowing down it, flowing back to their concentration gradient. They're trying to equalize. In the process, these protons keep pumping across. And so it's a constant movement. If you pump water up a hill and let it run back down and pump it back up and let it run back down, it's going to keep doing that forever. But we're creating usable energy. And it's extremely efficient. In fact, there's research being done into how can we possibly make hydrogen batteries and make them mass producible, because it's just incredibly efficient. The closest we've really gotten on a mass-produced scale is lithium batteries, which you see in, like, your, I think your iPhone, your smartphones have lithium batteries. Electric cars have lithium batteries, because lithium is also a very simple element, very low charge. Hydrogen is even more so, and so it makes really an amazing battery and energy exchange system. And so we need the only reason that this electron moves from this one down to this one, down to the next one, down to the next one, pumping it at the rate that it needs to. All these proton pumps are pumping because electrons are moving across them. That's all happening because of oxygen down here at this end. And it's pulling everything along because it's hungry. And so this one gets charged. And then the next one moves in and gets charged, and the next one moves in and gets charged. And this is why we need oxygen. We breathe in oxygen so that we can do aerobic metabolism. Aerobic means with oxygen. And so when we have this oxygen, we can do this whole part of breaking down food and make tons of ATP. It's just everything else looks negligible by comparison. So we get like a couple ATP out here and 2 out of here, right? So our total is like 4. Yeah, we have a net of 2 ATP here and here. So we have 4 ATP produced by just these 2 cycles alone. When we incorporate the oxidative phosphorylation, we jump up to these crazy numbers of like 36 to 38 per turn. Because what we're doing is the oxidative phosphorylation produces, on average, 32 ATP per turn. It's kind of like 32 to 34, which is why when we add this 4 ATP from the previous processes on top, we get something like 36 to 38. But this oxidative phosphorylation process right here is doing the majority of the work in charging batteries, essentially, for your body to use and be able to do all the stuff that you need to do, which is why if you suffocate, you die, because you can't produce enough ATP for your body functions to keep going on if you don't have the oxygen to pull this process along. So hopefully this gives you kind of a more comprehensible background on the subject of metabolism. We're breaking down glucose to essentially get ATP in an amount that we can use, a high enough concentration and rate to be able to do the things we need to do to keep living. And so we need a certain level of efficiency, we need a certain amount of oxygen, and all these intermediary steps are involved so that we can have this complex way of harnessing the immense amount of energy that's in the food that we eat in a way that's usable and not destructive to us. So yeah, that's pretty much it on metabolism for the first part. With part 2, if we do a part 2, we'll come back and use these base ideas to kind of explore some of the more specific ways that our body does metabolism.